The overall goal of this procedure is to synthesize, purify, and characterize lanthanide-containing complexes that can be used for tagging biological macromolecules. This is accomplished by first mixing the metal starting material and ligand while controlling the pH of the solution. The second step of the procedure is to purify the resulting complex using dialysis. Next, the absence of free metal needs to be verified. The final step of the procedure is the characterization of the properties of the complex that are relevant to imaging. Ultimately, results can be obtained that show that the synthesized complexes will behave as contrast agents through relaxivity measurements.
This method can help answer key questions in the imaging field, such as whether the water coordination number of a contrast agent changes upon binding to a protein.
Generally, individuals new to this method will struggle because not carefully controlling the pH will result in rendering the ligand useless for bioconjugation.
Demonstrating the dialysis and relaxivity measurements will be Buddhima and Sashi, graduate students from my laboratory.
To begin metalation using lanthanide salts, first dissolve 1 equivalent of ligand in water to produce a 30 to 265 millimolar solution. The ligand para-isothiocyanato-benzyl DTPA is used here at a concentration of 73 millimolar. Next, adjust the pH of the ligand solution to between 5.5 and 7 by adding 1 molar ammonium hydroxide. In this video, 0.2 milliliters of the 1 molar ammonium hydroxide solution is used. Now,